So here we are going to go over how proteins are marked for degradation. Because remember, in our body, we'll have specific proteins that must be degraded because, let's say, they've lost their function or they have been mutated. So the body must get rid of these dysfunctional proteins. But our cells must have a recognition process in place to understand which proteins are to be degraded and which are not to be degraded. And the way we can make note of which proteins are to be degraded, we can ubiquitinate those proteins. Now, ubiquitination is a process in which we take ubiquitin, which is a protein, and we attach it to our protein of interest. And this labels it for degradation. Now, there are 3 enzymes that are involved in ubiquitination. There's ubiquitin activating enzyme, ubiquitin conjugating enzyme, and ubiquitin ligating enzyme. And these 3 enzymes work in coordination with one another to add the ubiquitin to the protein we want to degrade. Now, the number of molecules of ubiquitin that we add to our protein can be variable, and the amount of ubiquitins that we add will have a different effect on the protein. A general rule of thumb to remember is that if we have around 4 ubiquitin proteins attached to our protein of interest, it will mark it down for degradation. Now, taking a look at the mechanism, activating enzyme comes in first. Now, this activating enzyme has this sulfur group, which is going to interact with the carboxylic group of our ubiquitin protein. This forms a thioester linkage. And the process of forming that thioester linkage requires ATP. And it actually requires 2 ATP equivalents because we are converting into AMP and pyrophosphate. Once we have this complex of the thioester linkage between the ubiquitin and the activating enzyme, we see the conjugating enzyme come into play. Essentially, the conjugating enzyme will replace the activating enzyme, or the activating enzyme actually passes the ubiquitin onto the conjugating enzyme. It's a passing game. So the activating enzyme will pass it on to the conjugating enzyme, and now the conjugating enzyme carries the ubiquitin through a sulfur once again through its cysteine amino acid residue. So once again, the ubiquitin has formed a bond with our second enzyme, and now what's going to happen is that the conjugating enzyme passes the ubiquitin onto our final enzyme, which is the ligating enzyme, and the ligating enzyme will transfer the ubiquitin onto our protein of interest. Specifically, the ligating enzyme will pass the ubiquitin onto a lysine residue of our protein of interest. To quickly recap one more time, first, the activating enzyme picks up the ubiquitin, it passes it to the conjugating enzyme passes it to the ligating enzyme, and the ligating enzyme will pass it down to the protein of interest. Now let's take a look at the bond formed. The bond formed over here is a pseudopeptide bond, and it is formed with the lysine residue and the ubiquitin. Why is it a pseudopeptide bond and not a regular bond? Let's take a look at the structure of lysine. So this is the structure of lysine, and this is our alpha carbon. Normally, when we form peptide bonds, it is with the amino end and a carboxylic end. So a true peptide bond should be over here. But it's a pseudopeptide bond because the carboxylic group of the ubiquitin is actually forming the peptide bond over here instead of over here. So this is why we call it a pseudopeptide bond, because the bond is not where it normally is found for peptide bonds. Now, once we've formed that linkage with ubiquitin, remember, the general rule of thumb is that we want 4 ubiquitin proteins bound to our desired protein to mark it down for degradation. So essentially, this process of ubiquitination is going to occur in rounds. So this was the first round, and now this process will occur again, and then it'll occur 2 more times until we have around 4 ubiquitin molecules. Once we have 4 ubiquitin proteins, we're marked down for degradation, and the protein will approach the proteasome. Now, the proteasome is a 26S complex comprising of alpha rings and beta rings, which make up the 20S subunit. And then we have the 19S regulatory particle up here and on the bottom. And overall, this is the 26S complex. Now, what happens is that the 19S regulatory particle is what recognizes the protein. It recognizes the protein. And the protein, it enters our 20S subunit. As it enters the 20S subunit, it will unwind. And we can see that over here. So the protein is unfolding as it enters the alpha and beta subunits of our complex. Now, the protease sites are actually found in the beta subunits of this complex. Once again, the 19S regulatory particle will serve as the recognition site, protein unfolds, and then it is cleaved in here. Once it is cleaved, it is going to exit the proteasome in smaller peptide chains and amino acids, and those smaller peptide chains can further be broken down by other enzymes. Now, what's important to note over here is that down here at the 19S regulatory particle, we have a special cleavage site for the pseudopeptide bonds because we want to recycle those ubiquitins for another round of ubiquitination. We don't want to lose these guys, so we have a special cleavage site for the pseudopeptide bonds. Now, there are 3 enzymatic activities that are possible, that are known in biochemistry for the complex. And those 3 enzymatic activities are that it is able to cleave after aromatic residues similar to chymotrypsin, and then it is also able to cleave after basic residues such like trypsin, and then it's also able to cleave after acidic and branched residues, which is like peptidyl-glutamyl cleavage. So the cleavages found in our proteasome are similar to these 3 enzyme cleavages. And that is essentially ubiquitination and protein degradation. The key thing to remember is that it's a coordination of 3 enzymes in a passing game with one another, passing on ubiquitin until it reaches the protein and marks it down for degradation.